Good evening, everyone, and thank you so much for joining us here at Breakthrough Cancer Research for our webinar, Pulsed Electric Fields for Cancer Treatment from Idea to Industry. My name is Orla Dolan and I'm the CEO of Breakthrough Cancer Research, and we're delighted to have with us this evening Dr. Declan Soden, the CEO of Mirai Medical. And so we know Dr. Soden very well. We've been watching his research over a number of years. And to introduce him to you this evening, he is a UCC graduate who first did a bachelor's in science in molecular biology, followed that up by a PhD in molecular genetics, and then was a principal investigator, and I won't give your age away, but for at least more than 10 years at Cork Cancer Research Centre, where you would have first encountered the technology you're going to be talking to us about today. And then from there, in more recent years, you've been able to spin out that technology into a company called Mirai Medical. And we're thrilled that you're going to be talking to us about that experience over a number of years. What I'm going to say for a little bit of housekeeping up the front before, Declan, I ask you to maybe share your presentation to give us some context to the discussion, is that we're inviting people to give their questions. If you can put them into the Q&A or the chat bot, and then we will pose them to Declan at the end. I know we have a mixed audience of both patients and researchers, so I'm sure we'll get a lot of interesting questions at the end. I do want to let people know that we are recording this, and so if there's any pertinent information that you don't want to share, hold your name back in the Q&A. So Declan, I'm going to invite you now to share your, oh look, I have one question already. Will the recording be available afterwards? And the answer to that is yes. It'll be on our social media over a couple of days. So Declan, look, I'm going to invite you to now share your screen, walk us through a little bit about your experience of taking this amazing technology and out from the lab into the industry. And we look forward to hearing from you. We'll talk to you again at the end.
Great.
Over to you.
Thanks a million, Orla, and thank you for the lovely introduction. Yeah, so that picture was taken a long time ago, to be honest. Yeah, I'm just going to share this evening my experience of developing this technology in the team we had at University College Cork, and supported by Breakthrough Cancer Research over the years, as we've kind of brought this out into to clinical studies and into CE approval and into the market, I guess, into hospitals around Europe. So it's been a pretty exciting journey, and delighted to have the opportunity to share that with you here this evening. So as already mentioned, my background, I have a PhD in molecular biology genetics, and I joined Jerry O'Sullivan, Professor Jerry O'Sullivan, the late Professor Jerry O'Sullivan, at the Cork Cancer Research Centre between the Mercy Hospital and UCC back 20 years ago. And that's where this story begins. So, maybe just to share with you initially, the mission for Mirai Medical, the company I founded, is to utilize this technology of electroporation, which I'll explain during this session, to improve the outcomes for cancer patients. That's our mission and goal as a company. And where it all started, where it began, I guess, if we go back 20 years ago, there was a paper published by the late Professor Jerry O'Sullivan, looking at the survival of cancer patients who had colorectal cancer, had surgery for colorectal cancer. And what himself and a number of pathologists and other researchers, including Professor Fergus Shanahan here in Cork, looked at was the degree of immune cell infiltration within the tumor and what that told them about long-term survival for those patients. And what was really interesting was that the patients that had more immune cells within their tumor, those patients tended to live the longest. The patients with the least number of immune cells within their tumor, this was the tumor that had been surgically resected, those were the patients that the disease tended to typically come back into. That data was reproduced at St. Mark's Hospital in London as well, pretty much identical data there. And they published a very nice paper 20 years ago on that observation, that the immune system and the engagement of immune cells within the tumor could predict the long-term survival of cancer patients. And that information, that research, very much drove Professor O'Sullivan and the research programs that we did. And when he hired me, when I joined his team, it was very much we were looking at how we could trigger an immune cell engagement into the tumor. So how could we make that happen? And that was our goal. And without being overly technical here, and apologies, some of these slides may be for some of the audience, but when you look at here's a tumor mass, essentially what we want is for the immune system to have access to the tumor. And one of the approaches we looked at early on was trying to introduce genetic material encoding for an immunotherapy directly into the tumor. And the way we did that or enabled that to happen was using a technology called pulsed electrical fields. So pulsed electrical fields is essentially when we apply a voltage for a brief period of time to tissue, and it renders the cell membrane porous. It creates a condition called electroporation of the tissue, makes it porous, allows us to introduce molecules directly in far more efficiently. So we could get DNA into the tumor cells like a Trojan horse, and then the DNA, once inside the tumor cell, can be expressed and we can have a therapeutic response. So we published a lot of really nice papers on this. It's really nice now to see with the COVID vaccines, with the mRNA vaccines, it's a kind of similar principle behind a lot of this. But back in the day, 15, 20 years ago, this was very novel, cutting edge area, and there is still tremendous potential for this technology to impact into the future for many cancer patients. But one of the observations we made, and we worked and collaborated with a number of centers in Europe, was that using the pulsed electrical fields to enable the uptake of molecules, some of the research in the Institut Gustave Roussy in Paris had seen that you could get chemotherapeutic drugs far more efficiently into tumors by delivering these pulsed electrical fields. Equally, more recently, some researchers have shown that we can introduce calcium more efficiently into the tumor cell and actually kill it off that way. So it's quite an effective way of triggering local tumor cell death. And one of the other key advantages of this idea of electrical pulses and delivering them to the tumor tissue is that the tumor is selectively more sensitive to the pulses. So we don't destroy the surrounding healthy tissue. We don't cause side effects. It doesn't have an associated toxicity with it. And so in a very simple way of describing this, here's your cell membrane, and we deliver the electrical pulses. It creates these nano-sized pores on the membrane of the cell, temporarily, typically. And healthy cells have a better capacity to recover from this. And certainly tumor cells are far more sensitive to this treatment, allows us to selectively target the cancer cells. So when you look at some of the data that we generated, and this is from Dr. Jim Clover's publication, a 5-year follow-up in patients treated with advanced basal cell carcinoma, we were able to demonstrate excellent responses and 85% curative outcome in the patients treated with pulsed electric fields or electroporation delivered in combination with low-dose chemotherapy. And you can see that in this case of a BCC on the eyelid, which would be a very complicated area to do plastic surgery on, an excellent overall response without damage to the underlying healthy tissue. And a similar example here as well of a patient who has a melanoma on their scalp, again, treated with these pulsed electrical fields. And typically this is done by using needle electrodes to penetrate the skin. You can actually see here in the middle image where those needle electrodes would have punctured the skin when the pulses were being delivered, but the scab and the damage is localized to the actual malignancy, to the melanoma itself. We don't see disruption of the skin in the area around it, and you can see the 3-month follow-up on that. So overall, very nice data, and I must say very proud to be part of a collaboration and a consortium between ourselves here in Cork and other centres in Europe. This was published, it's hard to believe now, 2006. This data was published. This was kind of based on our initial experience, and Professor O'Sullivan led the research on this. And I must say, it's fantastic to know that thousands of patients around Europe in particular are benefiting from this technology, or have benefited from this technology as a result of that development. And Dr. Jim Clover in Cork University Hospital, with the support of Alison Bracken Kennedy, continues to offer this technology to cancer patients. So it's a fantastic achievement. The fact that this immune response is triggered is also a very important observation as well, so that we see when we destroy or induce tumor cell death with this technology, we get an infiltration of immune cells, which is critical when we think about the first slide I presented in Professor O'Sullivan's observation that curative outcome is associated with immune cell infiltration. So we've published a number of nice papers over the years looking and clarifying, quantifying that response, and demonstrating that there's a number of important triggers induced when we treat tissue with pulsed electric fields that are very favorable, and which we believe there's huge potential for patients where we could potentially combine this with immunotherapy at the same time. So that ultimately led, the fact that we had some really nice results on the skin led to us developing the endoscopic system. So that led to the development of the Indove device. So a little animation here to kind of explain how this system works. So we have the ePore generator, which delivers these pulsed electrical fields. And we have the Indove device, which connects into that system. So what happens, I'll just speed it up a little bit. Here's the Indove, and it has electrodes either side of it and a little chamber. The endoscope is slid into contact there by the clinician, and then it's maneuvered into the gastrointestinal tract by the clinician to the area where the tumor is located. And then we use a vacuum to grip the tissue, pull it into contact with these electrodes. And the electrical field that's generated then encompasses the tissue around the device and kind of underneath it as well. That makes that tissue porous, as I showed you earlier, and that allows far better access to chemotherapeutic drugs and calcium directly into the tissue. So here you have the drug, here's our cell, the drug has poor access, we deliver the electrical pulses, and you get very nice perfusion of the drug into the cells after the treatment. The drug then delivers its effect, which in the case of some of these chemotherapeutic agents causes the breakup of the DNA within the tumor cells, and that forces it to die. So there's very little resistance that we're aware of to this particular approach to treating cancer cells. And then in the days and the weeks after the procedure, you get a very nice regression of the tumor. And then, you know, in some cases, a patient might require a second treatment subsequently, if not all of the disease was treated at the same time, at the point of treatment in the day. Okay, so we delivered this treatment to dogs, actually, believe it or not. Dogs get cancer too, and we treated a number of dogs with obstructive colorectal cancer. So this is an example of an obstructive adenocarcinoma in a dog that was obviously doing poorly at the time. And you can see at the follow-up at day 15, the obstruction is now cleared. There's still some residual disease present, which would have been retreated. And you can see it follow up a month later, the disease looks entirely gone, and again, completely at 235 days. So, you know, that's how you do your research. It started in the skin. We developed the endoscopic device and the system. We did our kind of validation work. We then validated it in dogs to show that it was safe, that we could deliver this safely. And that then allowed us to progress and do a clinical study in patients to show the patients with inoperable colorectal cancer could potentially benefit from this. Now, in a palliative setting where there weren't any other options. And so we had some really good data generated as part of this phase 1 study looking at patients with colorectal cancer, and that's again been subsequently published. And we saw nice responses in terms of patients' overall quality of life. And I must say, it's the rewarding part of the job is when you see patients who are in distress and suffering from a disease like this, to see their quality of life improve is a hugely rewarding part of this job. And here then that led from the colorectal study, we did a phase 1 study in esophageal cancer. And this is Mr. Michael Akim in Copenhagen University Hospital doing a procedure in a patient, again, with an inoperable esophageal cancer. And he's maneuvering the device into position to deliver the electrical pulses. And here's an example of an esophagus. Essentially, it's like a pipe that's obstructed. And you can see the before and after with the Indove treatment. And again, we've published that paper. So in terms of the timeline, I know the talk today is kind of around kind of bring the technology through and commercializing. And this is a straight line. I can tell you it wasn't a straight line. It was more 2 steps forward, 10 steps back at times, and a few cul-de-sacs along the way. But very thankful we've been able to pull an absolutely fantastic team of people together at Mirai Medical as we brought this technology out of the university kind of around 2018 or so. Obviously building on all that exciting work that was done over a 15-year period or so, and the exciting clinical data that we were able to generate. So we did a kind of a Series A investment in 2018, and that allowed us to kind of get CE approval, which allows us it allows for easier clinical rollout in Europe in particular, and sale of the technology. And then we also developed the QTIS device as well to allow us kind of better access to some of these skin cancers also. So last year, we treated our 500th patient. We're very excited about that. Obviously, things have been impacted with COVID over the last 12 months, but we were able to complete an investment round last year as well, which was fantastic. All done over Zoom like this, which is amazing. And then recently, in the last couple of weeks, we're very excited to be awarded a very significant grant by the Irish government, which is kind of peer reviewed by external international experts in cancer, which will support us to develop some new devices for gastrointestinal cancer to allow us to treat kind of a broader range of cancer indications from early to late stage. We've also just commenced the Vector study, which is a larger phase 2 multi-center study looking at esophageal gastric cancer treatment with Endove device. And we'll be launching the Pioneer registry to gather some of the clinical evidence being generated from all these patients being treated to look at the benefits of the system and capture it as well as we can. And then into the future, you know, we plan new devices, as mentioned, which will be supported by the DTIF grant. We're hopeful next year to kind of engage in the US as well. We have some exciting things happening there. And we're certainly hopeful to combine what we're doing with immunotherapy as well. I think there's one particularly exciting area that we're looking to investigate into as well. And so it's nice to say that research that began in Cork is spreading far and wide to cancer patients around the globe, primarily here in Europe at the moment, but more recently in Asia, and in a couple of months we'll be treating patients in Australia as well. And obviously then post-COVID, we hope to see this map expanding to patients across the globe as we can offer a kind of an innovative solution to patients, particularly those with kind of late stage gastrointestinal cancers. So kind of in summary, the technology we've developed, it can be delivered as an outpatient treatment, so it minimizes the amount of hospitalization time for the cancer patient. The side effects, you can never say there's no side effects, but the risk of side effects is relatively low for most of the patients that we've treated. The healthy tissue, as I mentioned, is preserved, which is a really important. So that means that and that's a really critical element. Where this technology is hugely beneficial is where the preservation of healthy tissue is crucial. So there's certain areas where that is particularly important, and that's a particular advantage of this technology. We see immune cell recruitment and an immune boost when we deliver the treatment. Because of the time in hospital is reduced and we're delivering this kind of as an endoscopic outpatient treatment, the cost is reduced. The electrical pulses, or these pulsed electrical fields, are non-toxic. And we believe we can treat all stages of the disease in time. At the moment, we're focusing more on the palliative end stage, but we feel there's room there for this technology to be applied to a wider range of cancers, and that's where this DTIF grant would help support us expand it. So at the moment, our focus, as mentioned with the QTIS device, is more for cutaneous disease, and with the Endove device for gastrointestinal cancers. We do have a very nice publication coming out with Professor Smith in London, looking at gynecological cancers treated with the technology as well, which is very exciting. And again, there's some other areas where the technology is particularly appropriate, again, where tissue preservation is important. There's benefits in delivering this system. So finally, just to say acknowledgements and thank you. None of this work would have been possible without a great number of people, and they're not all mentioned here on this list. First and foremost, Professor Jerry O'Sullivan, who gave me the tremendous opportunity to work in the Cancer Research Centre, to work with him and the team of people there, and to Breda O'Sullivan, his wife, very grateful for that opportunity. To great people there like Dr. Patrick Ford and Mr. James Clover and Alison Kennedy Bracken, the nurse, offering the electroporation service right now in CUH, and all the wonderful clinicians and scientists who've helped to kind of drive the cancer research space forward over the last 20 years. There's some really wonderful people in there. Finally, thanks to Orla and Owen for the wonderful work that they do in terms of trying to move the cancer research space forward and improve the outcome options for cancer patients. It's great to be part of something very positive. So thank you very much for the opportunity to share that. And happy to take any Q&A questions following on from it.
Well, I'm just going to introduce my colleague here as well, Dr. Francis Drummond, who's our research officer, who will be helping me curate some of the questions. And firstly, I want to say, look, on behalf of Breakthrough, thanks a million for taking us through a very whistle-stop tour through that journey, because I know, it's a huge amount of years of research, really, but it's wonderful to see a technology that in some ways we were an outpost for when it came in initially in kind of early, in the mid-90s, we'll say, or was it in the early 2000s, probably was the beginning of the ESOPI study. And now emerging out of Ireland, that technology is going across Europe, kind of with Cork and Ireland as the kind of place where it started. So I'm going to start with one or two questions that I have here and that had came into me by email in advance. And the first one is, why is cancer tissue more conductive than healthy tissue? You made reference to the fact that, it preserves healthy tissue, and that, even when the needles are around it, it really only affects the cancer tissue. Is there a reason behind that?
Yeah, I think the main reason is that if you look at the cancer tissue, the architecture of it, it's actually quite erratic or irregular. The lymphatic drainage out of that tissue is, generally speaking, quite poor, so there tends to be more fluid build up within it. Now, that said, I think there's probably there's other reasons as well in addition to that, because there's interesting studies being done. There's a company in the UK, Endoscopic, who are differentiating early stage cervical cancer by looking at the conductivity of pre-malignant tissue. And they're seeing a differentiator between malignant or pre-malignant tissue and healthy tissue as well. So there's an element of it. It's just the fact that the lymphatic drainage out of tumor tissue in general is poor. That builds up the amount of fluid, which changes its conductivity. And I think then more fundamentally, the actual cell biology of cancer cells, there's something different about them that you can actually measure that change between their resistance and healthy tissue that some researchers are looking into it as well.
But you've also looked at this obviously in skin, which, and then esophageal tissue, which is, probably more sensitive, and you're seeing that same effect regardless of location, that the difference between, like, obviously that it's selective in its action.
Correct. Exactly. Yeah, I mean, it's kind of fascinating. I wouldn't show all the images here. Obviously, some of them are part of clinical studies, but, there's a recent image of a patient treated in Spain with a gastric cancer. And you can see very much the area. It's just very specific where those pulsed electrical fields were applied. That's where the response is occurring. There's no damage to the surrounding healthy kind of gastric tissue. So in the stomach, in the esophagus, in the colon, we have a very nice, if I can kind of share, we have a webinar with Professor Ismail Gogonier next week for anybody. We can get the link for that, but he's sharing their experience with colorectal cancer in Denmark, which is really exciting. They're using calcium in combination with electrical pulses and Indove to treat patients. And that's really, really nice to see. But again, they're seeing the same sort of selectivity across the board. So yeah, it seems to be universal as far as we're aware.
All right. Well, before I pass it to Francis, you touched on something which was another question, and that is, you made a reference to the idea that, obviously you're making the tumors and the more porous for uptake of drugs, but also the calcium is kind of what you're substituting now sometimes for chemotherapy. And how is it that the calcium is working in place of a chemotherapy, in terms of killing the cancer cells?
Yeah, so kind of, I guess, if we go back a little bit, the idea of using pulsed electrical fields or electroporating cells, applying this voltage to them, that's been done in the lab since the 70s or 80s, and it's been used to kind of deliver genetic material into cells. It's been used for a variety of purposes in the lab. And then in the 90s, Dr. Louis Mir's group in Paris, they discovered actually, this could be used to deliver some chemotherapeutic agents far more effectively. And how they worked essentially is, well, particularly some of the drugs, they just bind to DNA. Once they bind to DNA, they break the DNA and the cell is forced into a cell death type pathway. So that's the kind of, that's the effect. You're making the chemotherapeutic far more effective. In the case of calcium, what's different is that the cell regulates the amount of calcium within it. It's very carefully regulated. It's about 10 to the minus 7 molar concentration within a cell. And so if you electroporate it and you've injected calcium solution beforehand, you get calcium being absorbed into the cell membrane reseals, and now it's left with far too much calcium within it. The cell then has to pump all that back out. And so it stresses the cancer cells. They've already lost a lot of their energy. And that's kind of another element to this, is that ATP, which is the energy store for the cell, they lose a lot of that in the process of being electroporated. And they then need that store in order, they need their ATP to pump the calcium to fix things. They don't have enough and it's lights out. So that's a very exciting possibility, to be able to inject calcium into a tumor, deliver these electrical pulses, and to see them shrink and respond to treatment very effectively. So we're really excited about the data that Professor Ismail Gaukenur in Denmark has been generating in his clinical studies. And like I said, there's a nice webinar on that next week for anybody who wants more of this.
We'll send the link out to everybody who attended, and we'll post it up on our social media if you share that with us. Frances, I'll hand it over to you for the next question.
Okay. I just had one before going into it regarding the calcium as well, when we're talking about it. Is there a difference in the immune reaction when there's calcium and when there's chemotherapy?
Yeah, there is. And so there was some nice work being done by Dr. Patrick Ford, looking at that, and actually by other researchers in Denmark as well, looking at what type of immune response is triggered. So yeah, the actual response varies depending on how you kill the cell. So let's say there's a particular way, if we induce cell death by having a chemotherapeutic agent there, that'll trigger a certain type of immunogenic cell death, it's called. If we use calcium, it changes. And it's hard to know which one might be better. Is there one way better than the other? I think obviously not having a chemotherapeutic agent would be preferable in a lot of senses, but that keep in mind the dosages of chemotherapy that are being used are actually greatly reduced. So we're not using conventional chemotherapy in the sense of patients get repeated doses multiple days over multiple weeks. They just get a single dose at a low level on the day of treatment. So there's research to be done to look at, I guess the next step here is combining this treatment with an immunotherapy and identifying, well, maybe the immunotherapy might work better if we use calcium, or it might work better if we use chemotherapy. That's something that needs to be investigated.
Yeah, really interesting. We have loads of questions here, Declan. So you talked about two mechanisms in fighting immune cells into the tumor and drugging the tumor more effectively. How do these two mechanisms compare in efficacy? And that's George Malarius. Apologies, George, if that's not how you pronounce your surname.
So yeah, obviously this is easier to maybe explain in a slide, but so keep in mind that, I mean, certainly as I understand this, and I'm open to correction, but as I understand it, okay, so we're saying chemotherapeutic agents have poor access to cancer cells, and that repeated doses are required as you get a chemotherapeutic treatment. But equally, as a tumor develops and evolves into a mass, access into it by immune cells in the immune system is actually quite limited. So you don't get a lot of infiltration occurring. So that's a problem. So on the one hand, while physically we're treating that tumor with these electrical pulses, we're causing it to break down. We're actually opening the doors up, and that allows the immune cells in. It's also, I'm trying to think the best way to explain this, but let's say at one level, we're affecting the cell itself. So right down to the cell, we're affecting that cell, we're making it porous, we're allowing molecules better access in. And then if we take a step back, we're also opening the tissue up, and that's allowing immune cells better access, and they can infiltrate into the tissue. And then to kind of really complicate things and go down another level, the actual type of cell death that we just spoke about that's triggered even the release of ATP out of the cell, the expression of heat shock proteins. There's a various numbers of stress factors that are triggered by this, which we have some nice publications on, which are called, they encourage immune cells to kind of say, come and have a look here. There's something to come and see. The missing piece is that it's the activation of those immune cells. So typically those immune cells would come in. We would hope they would pick up antigen from the tumor, which is mutated protein. We hope they would migrate then to a lymph node and activate the T and B cell response. But the problem is, and this is a problem for cancer patients in general, is that the cancer itself has depressed the immune system locally. So its ability to activate these T and B cells is somewhat diminished. It's depending on the stage of the disease. So that's a problem. The immune system becomes exhausted from trying to fight this tumor. And even though, say, with our treatment, we can tick a lot of boxes, but you need to tick all the boxes to really get an effective immune response. And the last piece is just the fact that the patient's immune system is so exhausted, you really don't generate that kind of systemic immune response as a result. So that's the really important piece that we're going to try and figure out next, as to how we can kind of overcome that problem, and hopefully by doing that help to improve kind of systemic immune responses and curative outcomes.
Very good. Yeah. If the same person says, great work, by the way, but also had another question. Do you think, or what do you think of tumor treating fields? Novocure.
Yeah, I've heard of that, tumor treating fields. It's the same principle, and it's been developed by a company in Israel. And it's a kind of cap that you put on the patient who has a glioblastoma, brain cancer. And essentially, the way that works, so they're applying kind of a low current over half a day. That's kind of in constant application for the patient. And it stops the chromosomes, as I understand it, that electrical field or tumor affecting fields, it stops the chromosomes from splitting. So it kind of stops the tumor cells from dividing and replicating. So, and the data from that is very exciting. It looks to be, the part of the challenge we have is the amount of clinical evidence required is very, very significant to really kind of make an imprint, to get people to kind of stand up and take notice and listen and say, oh, actually, this is really significant. I mean, on one hand, you're trying to provide evidence where there's 50 years of evidence, let's say, for this, the standard of care, the treatments that are there. And that's obviously correct. You have to do your trials, you have to provide the evidence, you have to do it properly. But one of the challenges with new technologies like this, even though it's exciting, is that you're kind of up against an almost insurmountable task to bring a new innovation through. But certainly the tumor fields treatment for glioblastoma looks very exciting, and their initial data looks really, really good.
Excellent. That's great news for everyone. William Gallagher says, great tour de force. Well done, Declan and team. Great to see the journey from discovery to clinical implementation. Just wondering if there are any possibilities to utilize this approach outside localized drug delivery.
So I'd have to have a chat with Bill directly on that.
So that's Liam, and he goes on. His next question was, what happens to the tumor vasculature following electroporation?
Yeah, it decays. So you have an interesting response where there's what's called a vascular lock immediately post procedure. So the capillaries shrink, the endothelial cells actually specifically respond to the electrical pulses in that way. And then what you see in the days and the weeks after procedure is that decays and that dissolves away. What's also interesting, it's worth mentioning, is that when we did some of this research back years ago, if you deliver this treatment to mice with a full immune system, you've got these curative outcomes. If you deliver the treatment to mice who had a portion of their immune system deleted, it didn't work half as well. So even though I'm trying to explain how it works, and we get the drug in, we kill the cells, like you say, does it affect the tumor vasculature? There's definitely, and even though I'm saying we don't create a systemic immune response because we don't generate at that level, there's definitely a very important element of the response that's kind of immune cell driven, that helps to clear the disease or the site in the area that we've treated.
And there's some evidence of it actually impacting metastasis, isn't there, Declan?
Yeah, I mean, we've seen some kind of, let's say, call them anecdotal cases, but we've certainly seen some really remarkable responses over the years in patients treated, where they've had systemic disease and it's completely responded elsewhere. But I'd say that's far more the exception than it would be the rule. But again, I truly believe that comes down to just the patient's immune response, and maybe sometimes it all lines up nicely, and we get a really good response overall. So look, the goal is to try and move that from being a rare occasion to being in the majority.
Absolutely. Elsie Jenkins says, wonderful talk, very exciting space. How do you think the translation of this technology to brain cancer would look?
Sure. Yeah. So like I say, I mean, the tumor affecting fields, that's been something that has been applied in that space, and I think quite successfully. At the end of the day, it comes down to designing an electrode that allows you the access to deliver the treatment. Obviously, into the brain is a tricky area, but there's no reason why. I mean, to Orla's question earlier, it seems to be specific in the GI space, whether we're treating gastric cancer, esophageal cancer, or breast cancer, or gynecological cancers, we're seeing the same results universally. So I would expect the same in brain. And really, it just would come down to developing the right type of electrode, and obviously working with the neurosurgeons and the specialists in that space to kind of develop that system to allow it for that treatment to be delivered. So it could be done, but as we know, from start to finish, as I just shared, yeah, it takes a long number of years.
Excellent.
I jump in with
on the 500
I'm going to jump in with a question that was sent to me by email, and that is, how does this compare, or is it the same as radio frequency ablation?
Yeah, that's a good question. So, if we look at the methods that are available to us to destroy a tumor, well, let's look at it, right? So you can surgically resect it. If you catch it early, the gold standard is to go in and surgically cut it out, get the margins, and that's great. If not, we go in with maybe chemotherapy, or clinicians go in with chemotherapy, or then we try to ablate the tumor. So radiation treatment, we're delivering radiation focused directly onto that tumor, but you're destroying it, we're ionizing radiation. The other options are things like radio frequency ablation, where you stick a probe into the tumor and you essentially heat it. So you create energy, you deliver heat into the tissue. There's cryoablation, where you freeze the tumor. There is microwave ablation, same idea. In a more targeted sense, you're delivering heat to the tissue. What's different with what we're doing is, though, we're generating these pulsed electrical fields. They're only happening for a millionth of a second, when we're kind of spreading it out over a few seconds. And we're essentially not looking to heat the tissue at all, even though there may be some localized heating around the site of the electrode. But if we were heating the tissue, we would damage the healthy tissue structures as well, which we're looking to avoid. We're essentially just making the cells within that electrical field porous, and that allows us to kind of get these therapeutic agents in far more effectively. The other side of it as well is that we can just deliver more pulses, and these cells, the pores never seal, and it's called irreversible electroporation. And that can be effective without any drug as well. So patients with pancreatic cancer are benefiting from that. There's a company called Angiodynamics doing NanoKnife, which is essentially the same thing. They're just delivering a lot of pulses directly into the tissue.
And there was a question from Edmund Flynn, which was kind of related to that. How long does the cell stay more porous after the electrode treatment? So in your case, you're expecting it to close back up again. And what's the reasoning for wanting it to close back up again? And then how long? How long is it? Like a microsecond, an hour? Do you know what I mean?
Yeah. So the cells start reseeding pretty much immediately. Healthy cells, maybe important to clarify, I believe healthy cells in the vicinity of the electrical pulse are going to be affected as well. They're going to be porous for a period of time. They have a better capacity to recover, we believe, and so they're going to seal up quicker. So in the testing, we actually looked at this before, up to about an hour. So let's say for cells, and it depends on the pulse parameters that you use, but from about 5 minutes on, you start to see them sealing up again. And that kind of reaches a maximum at about an hour after the treatment. If we delivered more of these electrical pulses, that resealing would never happen. and the cells would stay open, and that would induce them to die kind of in the days and the weeks after a procedure. So it kind of depends, but the reason why we want the pores to, or that we wanted to seal up, I guess, is that we wanted a very localized treatment. You have to be, I guess, careful in that, you know, just going in and launching, you don't want to trigger a massive inflammatory response within the patient's tumor. So if you just went in hard and, you know, really kind of delivered too many pulses and just kept on zapping, wouldn't that be great? But it wouldn't really, because you'd create this massive inflammatory response, which would be negative for the patient. So by kind of doing it in a gentler sense and just kind of making that tissue porous, you allow the drugs in and they can do their effect, whether that's a chemotherapeutic agent or a calcium level. You know, you've got this very nice localized response, which, if you allow the immune system then in to kind of better recognize the tumor associated antigens, that gives the patient hopefully the best possible outcome.
Do you want
Yeah,
to?
and Hurley there asks, is the measurement of the tumor important? And also, if the cancer is genetic, would that rule out this specific treatment?
So the last question, no. I mean, it wouldn't really matter whether the cancer was a genetic inherited disease, not at all. There's no particular reason why that would be the case. And in terms of the number, is that the size of the cancer?
Sure, I think that was the question Anne was asking. Yeah.
Yeah. So obviously, the bigger a cancer is, the harder or the longer the procedure, the more pulses will be required. But again, what's advantageous here is that there's no reason you couldn't come back in 6 weeks or 8 weeks time and treat it. And we've done that with some of the GI cases where there's been large obstructive disease, and we followed up a treatment again kind of 8 weeks later. So, you know, if it shrinks down 50% after the single treatment, and that, you know, at the end of the day, this comes down to patients' quality of life. If you're doing this without impacting on their quality of life, you know, you can do it a second time, you could do it a third time if necessary. So some of Mr. Clover's patients in CUH would have had, you know, malignant melanoma, and it keeps recurring. So you treat a site, it responds there, it pops up somewhere else, and it's a very difficult cases that he treats. But, you know, there's nothing he can treat, and it might be 6 months later, it might be 3 months later, it might be 2 years later. So there's no reason why a second treatment can't be delivered if necessary, or if it's a large tumor that, you know, a second application couldn't be delivered.
Yeah, and I suppose with cancers, there is a dysregulation of the genetics, so it doesn't matter that it's a genetic based cancer.
Yeah, exactly. So in the case of this technology, it's kind of agnostic to the type of cancer. So what we've seen is, regardless of histological type of cancer or genetic mutation, we're not. So for instance, if we were putting calcium in, it doesn't matter what genetic mutation that, or what the origin of that tumor is. Universally, cells can't tolerate having excessive amounts of calcium internally. Similarly, if we use bleomycin as a drug, its mode of mechanism is not a cellular pathway. It actually binds to DNA and breaks it. So, and it does it very effectively at low doses. So typically in our experience, the histology type of cancer, it's kind of irrelevant. Where the challenge becomes is more getting the electrodes to the site, you know, being able to apply it to all of the disease. And the other challenge is obviously building up the clinical evidence to back up. You know, it can be wonderful to present this, but, you know, you really just need very significant amount of clinical evidence to change the clinical pathway.
So Sharon McKenna, great presentation, Declan. Proud to see this technology advance so far. Fantastic option for treating esophageal patients who currently have so few options. But would there be an option to develop this with the new RNA molecules currently in COVID vaccines?
Yeah, it's an interesting question, Sharon. And, you know, ultimately ultimately, you're trying to improve access. So I guess, you know, when it comes to treating cancers, and we're trying, there's a physical barrier there of actually getting the molecule you have into the tumor cell. And so, you know, if we look at the new vaccines, there's obviously some safety concerns, it's very small safety concerns around some of them, but that's just the molecules, isn't it, or the packaging that they may have. And so you want to kind of enable and make it easy for the RNA to get access into the cell. Once it's in the cell, it can deliver its therapeutic effect. So there's no reason why you couldn't electroporate that tissue to be targeted and co-inject the RNA in at the same time. So if you've got the right electrode, there's no reason why not. Happy to collaborate with you on that, Sharon.
I have two quick ones as well from me, which is you referenced kind of in the earlier part that say in Denmark, they were giving this treatment to patients that were unsuitable for surgery. So it was a really good option, as Sharon mentioned, for people why was it that they might be unsuitable for surgery? Would it be comorbidities, age? Is there a reason, the size of their tumor, or what was the reason why those patients might be surgical wasn't an option?
Yeah, I think, you know, some of that would come down to age and respiratory issues. Could be any other kind of list of comorbidities as we fail as we get older. You know, and you know, obviously, if you're going in, you have to have general anesthesia. A lot of patients can have that, as it can be a problem for them. So any number of other health related issues. Some patients aren't able to tolerate chemoradiation treatment as well, aren't able to kind of tolerate the full cycle of treatment. Yeah, there's kind of a variety. Some patients may have had surgery already. Again, surgery isn't offered unless you can get to all of the tumor. There's no point just taking out half it. So in a lot of cases, that would be an important point. It's just maybe it's around an important vascular structure which can't be surgically removed. That makes it inoperable. So again, we've done plenty of cases where the tumor is located next to an important vascular structure, and thankfully, yeah, it hasn't created any complications. We've been able to deliver the treatment.
So you said then in your pathway that you got CE approval in Europe. And
sure.
what's CE approval and why was it important? Was it really hard to get? You know?
Yeah, it was certainly. I mean, so the regulations in Europe for medical devices, there's the Medical Device Directive, and essentially the CE, or Certificate European. You see it on different products, don't you? But obviously, when it comes to medical devices, and this is kind of an important part as well of why you would commercialize or bring this technology out, in that you need the company that's producing this product to have a quality system, an ISO 13485 approved quality system. And then you need to submit your technical files to a notified body, all of the evidence, all of the data to support the use and the safety of the technology. It's kind of different in America with the FDA. You do this in parallel with building the clinical evidence. In Europe, it's kind of slightly different, although we're moving kind of to kind of probably a middle ground with FDA, with the new medical device regulations that are coming into place this year. So yeah, it's essentially having the CE mark simplifies, in some respects, it then becomes a clinical decision to use the technology. So it means you don't necessarily have to go in there and do it as part of a clinical trial. It can be a decision to, is this appropriate for this group of patients? And yeah, it is hard. I would say it's been the one part that's probably surprised me the most in terms of the workload and the demands. I would say half, if not more, of the activity of us as a company is quality and regulatory. The paperwork, we call it the bureaucracy. It's important, don't get me wrong, but it actually is an increasing burden on small companies. I see it's challenging to bring innovation through unless you have raised a very significant sum of money, you know, to kind of gather that data, gather that team, and bring it all the way through. But look, you know, there's a balance there between protecting patients and obviously making sure quality product is delivered to the marketplace. And hopefully, you know, we've been lucky with this DTIF grant recently to support you to kind of develop new innovation as well and bring that through into the market.
Do you mind if I ask one more before I hand over to you, Francis? So look, in your pathway, right? So obviously Breakthrough are like really proud of being involved in supporting a lot of this technology in the early, like all the kind of preclinical work, and we're supporting work in different parts of Europe on it still. And then, you know, it becomes, you know, you do trials and then you've got to take it out as a company and actually commercialize it, because the latter stages of trials are so expensive. But when you were getting support other than charitable support, particularly for researchers who are listening, what other type of support did you get from the Irish government? Or like, because you need to inject, you know, funding into this to keep it going. And obviously as a company, it's just developing. What other places did you secure funding from over the years?
anywhere and everywhere. Any grant that was I mean, I must say, like all of them, but Enterprise Ireland in particular have been hugely helpful over the years to help us to kind of develop the technology. They would be the ones who've kind of invested the most through grants through the university, and then actually grants into the company as well. They've been really, really big supporters of this. And, you know, it's an easy one to explain the benefits, and it's an easy one to kind of get the point across. But yeah, EI have in particular been huge supporters. But you know, back through the years, SFI, HRB, I think it's all changed. The EU, you know, but like, yeah, everybody, Esophageal Cancer Fund as well with Noel Ryan was hugely helpful. So yeah, any grant that was going, we applied for them over the years, and yeah, they were all welcome. And
an opportunity beggar.
exactly, that's it, you know. So yeah, very grateful to all of them.
And did you have to secure funding for, like, say, the treatment in Australia, Asia, States? You know, what's the pathway?
What's the deal? Yeah, so in the case of Australia, we're working with a company there to help introduce it. So we've kind of gone through, so having the CE approval streamlines it. So we have registration approval in Australia now, which is great. So we'll be doing training virtually like this. Obviously, no plans to travel to Australia in the next couple of months, but we're hopeful that patients over in Australia will start to see treatments being carried out there in the coming few months. So yeah, I mean, it's actually reasonably, I would say reasonably straightforward, but it's kind of, yeah, it's once you have the CE approval, that certainly helps things. The same in Singapore, same in some of the countries in Asia. If you have the CE mark, it really helps to kind of streamline getting the necessary registration and approval. It's different than if you don't have that, everything has to be done in a clinical trial. And obviously there's just, again, the challenge there is just the time involved. You can almost become out of date by the time your trial starts. But yeah, at least if you have the CE approval and you have some papers published, that makes the bureaucracy for the clinician just simpler, that they don't have to go through a trial network. They can just they can try and identify the right patients and offer it to them.
Yeah, or whatever process is there, exactly. One last question, if Orla and myself actually managed to be quiet. Your presentation mentions pancreatic cancer as a future target. What are the current barriers to bring this technology to patients with pancreatic cancer, and how could we speed up this process for the patients who need them?
Sure. Well, there's some really exciting work being done by the Angiodynamics group in the US and the NanoKnife system. It's the same principle. They're applying a lot, but they're doing this without drugs, and they're just delivering a lot more electrical pulses. And I know they have a study, the DIRECT study, which is evaluating this technology in pancreatic cancer. And again, you know, the publications coming out of those studies are very exciting. I mean, I think I saw the doubling of life expectancy from patients treated. So they're now doing a trial where patients are getting chemotherapy alone or chemotherapy plus this NanoKnife system to see, does it kind of improve their life expectancy. But I think the challenge really is just, you're up against, let's say, this is kind of a clinical trial discussion, isn't it? So there's so much data required to kind of shift the needle in terms of getting people's attentions, oncologists and other clinicians. And, you know, if you're a big pharma company, they tend to do studies in the hundreds, if not thousands of patients across multiple jurisdictions that costs millions and millions of euros. That can be quite challenging to replicate for medical device companies, quite time consuming. And you're not talking about the same sort of reimbursement rates, you know, between an oncology drug versus a medical device. So the kind of investment isn't there. It's kind of it's a problem, actually, to be fair, I think, in the cancer space that needs to be tried to be resolved. But yeah, that's the challenge, really. So going back to the question in pancreatic cancer, yeah, there's the NanoKnife system. It's there. There's some really good results with it. They're doing a trial now in patients with chemotherapy and NanoKnife treatment. So look, hopefully that delivers some really positive results in the next year or two.
Okay, I will stop.
And so I've got two little final ones for me. And one is, and I think Francis kind of mentioned it before, we did hear previously that, like, at times you can treat one tumor, like a primary maybe here, like on somebody's arm who's got melanoma, but then another site, like on their lower leg, might still shrink, even though it wasn't treated at the time. And I think it was some fancy word like an abscopal effect or something like that. But is that due? So is that why you're so kind of excited about, obviously you need another thing to come in to try and boost the immune system, but is that why, is that where the theory kind of came from? Or is it all about antigen presentation? And
yeah, so I mean, if you can treat a primary tumor, so you know, I guess the thinking or the logic here is that, you know, a patient has a cancer. By the time it's detectable, there's hundreds, if not thousands, if not hundreds of thousands of cancer cells circulating. So even if you may not see the second site or where it is, chances are it's out there. And what cures patients ultimately is the immune system recognizing those cancer cells or something specific to them and destroying them, or never allowing them to develop into a secondary. So so that's the kind of the logic. And so, you know, I always hear surgery is curative. And to all my surgical friends, I say, well, maybe, but it's the immune system is the curative component. They do their really important job of cutting out the mass, but it's the immune system which what offers the curative component to the treatment. And so, you know, anything we can do to treat the primary that helps the immune system to engage. And again, there's some really nice data being published on using immunogene therapy. What surprises me the most is actually immunogene therapy not having been translated into kind of mainframe application up to this point in time, because the data from it that I've seen is really, really compelling. You know, IL-12 has been delivered as a gene into patients with melanoma and combined with anti-PD-1 immunotherapy with some really, really good responses, which is like to the abscopal effect that they've seen, like they treat a primary lesion and all the other lesions just literally melt away. So, you know, there's definitely, there's an hopefully over the next couple of years, we'll see, you know, that technology, because I don't see there's any reason why it shouldn't become more mainstream and available to more and more cancer patients. And equally, I think there's a responsibility on us to try and get the cost of cancer care into a more reasonable level for patients and healthcare systems. It's kind of gone astronomical, the cost of care. And I think immunogene therapy could potentially offer an exciting way to reduce that cost, simplify it, reduce side effects for patients, so kind of a far more targeted treatment to them. So look, it's an area we'll be certainly looking at and trying to work on. And maybe, I'm not sure, 20 more years. I'd like to think maybe 5 to 10 years, but we'll see. Hopefully that come to fruition.
We'll check back in. Listen, you've been very kind to give us so much of your time. And so I'm going to just leave by asking you one final question, because we do have, and this is so you can conclude for us. And that is, look, you've been kind of in the trenches with the technology that obviously you were inspired by to take it on and to go into different directions and create devices. What are you most excited about in your area for the next few years? Like, you know, what should we keep our ear to the ground about that you're working on, that you're most excited about going forward?
Yeah, I love these big questions, Orla.
I'll give you an hour. No.
I know. Yeah. So, I mean, I guess more of the same of what we just spoke about over the last half an hour, really, and particularly with the immune response. I think, you know, in cancer in general, like I say, go back to that publication Jerry made with Fergus back, you know, 20 years ago, that the immune response is and it plays an important role. You know, that was kind of ignored and not really much attention paid to it. It was kind of pioneering at the time. And, you know, it's taken a long time for that to kind of to shake things up a little bit. And now over the last number of years, it's been really exciting to see some of these new immunotherapeutic drugs come through and to see the impact they've had in so many cancer patients' lives. I'd be excited to see what we're doing, kind of helping that or playing kind of a small cog in the overall wheel of that activity. I'd love to see the technology we have been able to boost the immune response, obviously improve patients' quality of life and shrink their tumors, but to be used in conjunction with an immunotherapy or an immunogene therapy to offer patients a curative outcome. I think that's really, really exciting. So it excited me 20 years ago. I don't get bored easily, obviously. So yeah, I think, you know, there's tremendous potential for that area to impact positively for cancer patients. So look, hopefully we play a part in helping to bring that through.
Well, look, we wish you the best of luck. I mean, we're proud to have been big supporters of this idea for a long period of time. And I hope it shows some of the people listening two things. One, which is why it's so incredibly important to support research at the early stages, so that those ideas can then translate out. And to also to know that there's really committed people who are trying to find ideas for cancer patients all the time, and that they have the same drive and enthusiasm that you have to see it come through and patients actually benefit. And we really thank you for your time today and for sharing the story with us. So
a pleasure. Thanks, Orla. Thanks, Francis. Thanks, everyone. And look, yeah.
Good night. Good night, everyone. Thank you.
Talk all night.